Hi, my name is Neko Okafor, and this is my extra credit for Intro to Biochem, the visual arts medium. I'm in section 2. Think back to the time when you liked to play. You took ordinary objects and clipped them away. Paper clips connected and hooking them together made things more unison and look even better. Similarly, amino acids are the building blocks of proteins. They come together to form different types of peptides and things. So today I'm going to talk to you about forming of these bonds between amino acids, and I will also talk about the different molecules that can be made in making it fantastic. In our cells, amino acids come together. In return, it creates peptide bonds called ribosomes and whatsoever. But we don't have to worry about that right now. Let's try to figure out why this happens and how. Let's take two amino acids called glycine and alanine. Let's see what forms when we connect them by any means. I decided to draw the glycine on the left and alanine on the right. The side with the free amino acid is the N-terminus, and the side with the free carboxylic acid is the C-terminus. If glycine is on the left and alanine is on the right, when they come together they form a dipeptide. The way this happens is that the nitrogen on the amino acid group of the alanine forms, which creates a bond with the carbonyl carbon of the carboxylic acid on the glycine it performs. The byproduct of this reaction is water, which means that this is a dehydration reaction, loss of water, but let's not squatter. Back to what we were saying before. This results in a molecule that forms a dipeptide or peptide like I said before. It will also be possible for this dipeptide to come into contact with another amino acid such like serine. And if this happens, then it is possible that it could form a tripeptide and become amine. This is done by if the nitrogen of the serine amino acid is being bonded to a carbonyl carbon of the carboxylic acid. Now that we got through what peptides can form, did you know that H2O can be a byproduct of a dehydration reaction? What a deform. If this actually happens, we would be left with a tripeptide, consisting of glycine and alanine and serine which fastens. So while it's pretty cool to see a tripeptide in all of its chemical glory, let's not forget how we write the peptides in a story. In this case, glycine and alanine dipeptide, we can write them as two circles that join together and do not subside. This is that right here. It will almost look like those paper clips I mentioned before. See what I tell you? Everything I'm saying connects and becomes galore. So back to what I was saying on how to write a peptide. Sorry I got off topic, but now I'll begin to recite. With the short names of amino acid inside, in this case is gly and ala. And now let's confide. If we were to add the serine from the tripeptide, they would form a glycerol, oh my. So think of this as a good way to represent a peptide, where the free amino acid is on the left-hand side, and the carboxylic acid is on the right-hand side. Well, that completes my lesson on peptides and protein building. Hope this helps and keeps the studying moving.